welcome, and it's a real pleasure to be able to share with you some of our work on the topic of advancing genomic medicine through global collaboration. So my work focuses on deciphering the causes of rare disease, and the challenge in our field is that about 60% of rare disease cases remain unsolved. And for that reason, Daniel MacArthur and I started our Broad Center for Mendelian Genomics, as well as the Rare Genomes Project, to try to address this. So the center is now under the Gregor Consortium, and it's led by Anna Donna-Luria, Mike Talkowski, and myself. And we're really trying to apply novel approaches to solve the unsolved, which is true of the entire consortium, new methods, new strategies. Over the years, we have collaborated with many amazing investigators shown here on this slide, and collected thousands of samples from collaborators all around the world. And that, to date, already has led to nearly 1,000 novel gene discoveries or candidates identified. About a third of them are well-supported, publishable results, and about 2/3 are still in that candidate stage, looking for added evidence to prove causality. So we also have our own direct recruitment arm called the Rare Genomes Project. And in this study, we're particularly applying whole genome sequencing to solve rare disease, whereas most of our other collaborations, we usually start with exome. Even if they've already had an exome, we have better methods today than perhaps were applied years ago. But with this study, we've really directly applied whole genome sequencing. We've looked at over 1,000 families are enrolled, 600 of which we've analyzed, and we've been able to identify returnable results in 149 to date. All of those orange dots show those families that we've returned results, despite the fact that many of these families have been on multi-year diagnostic odysseys. Now, we did look specifically at that genome data to say, well, how many of the discoveries that we made actually required whole genome data versus an exome? It's an ongoing question in our field. Is it worth the extra money? And what we found is that if you look carefully, at whether you could have solved that genome with an exome, we found that the increased diagnostic rate was about 12%. And a lot of those were structural variants that weren't easily detectable by exome sequencing, even using the newest methods of CNV analysis. But also really being able to look into non-coding region. In particular, I'm going to talk a little bit about analysis of deep intronic variants. So in this Rare Genomes Project case, 1477, this patient had developmental delay at 6 months, infantile spasms. On MRI, showed lissencephaly, and they had negative gene testing, both panel, microarray, and exome. And this family just really wanted to understand what was going on, as well as understand risk for future children. So we were able to look, as I said, we did a genome analysis and look deep into the introns as well. And what we identified was a deep intronic variant, about 3,000 bases into the intron, and Splice AI, which is a machine learning developed tool to better detect both disruption of existing splice sites, but also the creation of new cryptic splice sites. And this was predicting a gain of an acceptor site, which was in the DCX gene, an X-linked cause of lissencephaly, and the physician of this family really felt that this was a good diagnostic match. We are following up with functional analysis of the splicing effect, but we think this is a pretty good diagnosis. And it really shows the power of more sophisticated AI-based splicing analysis approaches that can really pull the needle out of the haystack, shall we say, to identify novel causes of disease. Now, sometimes if we're dealing with diseases where we can get access to tissue, it makes it easier to quickly validate those results, or even pick out results we might not have looked at otherwise. So in this case, this is a baby with profound hypotonia, weakness, ptosis, and reflux. And we were able to get a RNA-seq analysis on patient muscle tissue to add to our genome analysis. And in doing so, and this is just one of these volcano plots looking at expression of genes, what you can see is the most distinct outlier in this expression analysis was the MTM1 gene, a muscle gene. Also looking at splicing, you can see that in this top orange arc is basically altered splicing in the proband in this particular gene. And with that signal through these techniques, we then could zero in on this region, and in doing so identified a retrotransposon line 1 element insertion into intron 3. The mother was actually mosaic for this variant, which suggests some recurrence risk issues as well. But this allowed us to make a clear diagnosis by combining the genome data with RNA-seq and together make a very definitive identification of causality. Now, all of these tools and approaches that we use are available to the community in our open source software called Seeker that allows for exome and genome analysis. We've now put it on the NHGRI funded Anvil cloud-based platform, so it's freely available to anyone to use. And we actually have over 2,000 investigators from over 100 countries that have performed analysis using our Seeker platform. We have YouTube videos and other ways to train people how to use this. So we really encourage you, if you need a free platform for genomic analysis, to investigate Seeker. Now, another aspect of the work we do is recognizing that most rare diseases are incredibly rare, and there's no single investigator often amasses more than one case for a particular disorder, as we're, beyond the low-hanging fruit and now dealing with the really, really rare stuff. And solving these disorders will require immense data sharing, and that is another huge strategy that we focus on in our rare disease program. So I'm going to give you an example of that, the Rare Genomes Project Case 434. This was 2 SIBs with microcephaly, seizures, global developmental delay, and they were non-ambulatory. And we found 2 novel variants in the PPIL1 gene, which had never been linked to the disease at the time that this case was analyzed. One variant was a nonsense variant, so more suspicious, but the other was the run-of-the-mill missense variant, not that suspicious. So it was a candidate, but certainly not enough evidence to implicate this gene in disease. And it's a challenge. It's been a challenge to our field, because years ago we had this common problem. We'd have all these gene candidates just sitting on the shelf, never to be published, waiting for more evidence. But when these diseases are really, really rare, an individual investigator may never get that evidence. So what did we do? We gathered a community. We came up with this concept, genomic matchmaking, where an investigator has a candidate. Could they put it into a system and match with someone else around the world that has the same gene candidate, and then allow those phenotypes to be compared to look for commonalities in phenotype to validate that match? So we worked together. We gathered at the ASHG meeting in 2013. We defined our goal, really gene matching. We defined the barriers. People didn't want to get scooped with their gene candidates. So how can we do a safe way to share data, bring evidence together, and co-publish? And to do that, we defined all the building blocks, the data models, the APIs, the rules of engagement for connecting to databases, the consent requirements. We did this in conjunction with the Global Alliance, where we utilized the different work streams of the Global Alliance to develop these different approaches. And then in 2014, just a year later, we connected the first 3 databases and have been building the database ever since. It now has 9 different nodes connecting databases around the world. And this is probably one of the first federated genomic data sharing platforms, because everybody's data sits in their own databases, does not need to be centrally collected, but all the databases allow these queries to do gene by gene matching. And in fact, this is, I would argue, been the single most successful tool we've had to contribute to rare disease gene discovery, is this data sharing platform. There's over 15,000 candidate genes from over 200,000 individuals, from 13,000 contributors from 103 countries. And in the recent review we wrote, over 1,000 novel genes have been discovered using this platform. And that's just looking at 3 sites that tracked their data, 3 clinical labs. I believe there's probably more than this that's been discovered. So a really powerful tool to do this. And if we go back to that case I mentioned earlier with the PPIL1 gene, when we put that gene candidate into Matchmaker Exchange, we immediately matched with 7 other families, and were able to join with a study that was led by Joe Gleason, who also made a mouse model of this particular gene, really solidifying this gene's role in disease. However, we do have a challenge in that not every case do we identify a candidate. So how do we, solve the cases that have no candidates? So, and this is not just a challenge in in gene discovery, discovering novel genetic cause of disease. But in routine genetic testing, we have a massive challenge as well, in that most of the variants we identify we can't interpret. 41% of all variants in ClinVar are VUSs or conflicting. And in fact, in a recent study we did looking at genetic testing in North America, looking at 1.5 million tests offered to patients, what we found is that 30% of all genetic test results yield an inconclusive result due to a VUS. So this is a huge challenge our community is dealing with. So to solve this, we've moved on to the next phase of what we're trying to do, which is what we call one-sided matching. In this case, maybe you have a variant that looks a little suspicious. It might not even raise to the level of you're willing to put the gene into Matchmaker Exchange, or it's a known gene. It's just simply a variant you don't understand. But what you'd like to do is query every genome in the world to see who else has this variant, and then get phenotype returned from that any case with the variant. So this is what we're working on now with the Global Alliance. And we've developed a list of different groups that are working hard to really start to put the pieces of the puzzle together and essentially build another federated network, but this one really centered around variant level matching and querying. Before, you had to have a case to query this network. I'd like to see more range of use cases here where you can have a suspicion about a variant and simply query a database, not necessarily have to get an email every time there's a match. So different ways we're likely to expand this network, but still focus on the concept of federated data sharing where the data stays at the organization, but we can query using single variants and return lists of genomes with their phenotypes. And we've been working with the Global Alliance on some of the new APIs that have been developed. Right now, we're really focusing on the Beacon version 2 API to really drive this network. And in fact, there's already been use cases developed so that Johns Hopkins team that brought you Gene Matcher now has a Variant Matcher, and anyone can register an account with that database and search, and if you put in a variant with the phenotype you found it in, they will return a list of all the cases they have with that variant and their phenotypes. And it's become very powerful, but it's just a single node right now. Now they're working to connect to other nodes in this sort of federated approach. Likewise, the Beyond 1 Million Genomes project in Europe is building this connectivity through 26 European countries that have genomic data to essentially enable access and query of that data using this same Beacon version 2 API. And so we're really working to get this community together to advance this network, which I think will be critical in this next phase of interpreting variation and gene discovery. Now, I was asked to talk a little bit about the consent, what is required in terms of using these systems and sharing data. And I want to point out that we've, for a while now, really established that consent is not required for sharing variant classifications with summarized evidence in the context of clinical genetic testing. And that on this left side of this figure sort of shows when we bring aggregate evidence together on a variant. You're not necessarily showing individual level data like on the right, where it's a female, age 50, with this ancestry and this phenotype, and do they have any other variants? That is more detailed individual level data. It does require consent, but this aggregate evidence sharing, even if it's just one case, is really okay to be able to submit. It's your claim on a variant with the supporting evidence. And we have more information and videos that we've created in different languages to explain to patients and families what the difference here is. And we wrote a paper to really solidify this, this consent requirements for case level sharing versus just aggregate evidence. Also in the UK, a group of colleagues really wrote up a similar analysis saying this consent is not required if you're sharing this aggregate evidence, and that this is consistent with the GDPR requirements as well. So both of these really helped enable clinical labs to feel comfortable sharing data into ClinVar. And here's an example of just that sort of aggregate evidence you often see in ClinVar, where they're summarizing all of their data on and to justify a claim of pathogenicity or claim of being benign. On the other hand, we've put out a one-page consent form. If you simply are a physician, you want to help consent your patient to share more detailed data, which is very useful to us, to have more detailed phenotype, as we talked about before. And so if you can consent the patient, terrific, then you can share more detail. And here is an example from our Genome Connect patient registry, where in our entry into ClinVar from this patient registry, you can see really detailed phenotypic data, age, gender, all of that information connected to this case. That can be very helpful in deciphering whether these variants are causal or not as you aggregate submissions from different labs. So two different types of data sharing, one requiring consent from the patient, one not. We also went through this process thinking about the use of the Matchmaker Exchange platform. And in this case, the level 1 sharing, where we say it's just structured phenotype and a candidate gene name, no additional consent is required in the clinical care setting. But it is worth pointing out that if you're doing research, then this type of use case should have been part of your research consent. If you go to level 2 and you're really getting more detailed, unique, or sensitive phenotypic data, then consent would be required either in the clinical setting or certainly in the research setting as well. So this is sort of the paradigm that we've been working under. And now we're working to really define this for this variant level matching, where we're querying raw genomic data sets and really thinking about what kind of access and consent requirements are there. And we're working with the Global Alliance in really developing this framework better. And with that, I just want to acknowledge a number of the projects I talked about, lots of people involved, our Broad Center for Mendelian Genomics, the Rare Genomes Project, all part of the Gregor Consortium, the Global Alliance for Genomics and Health, Matchmaker Exchange, ClinGen, ClinVar, and my team at the Broad, the Translational Genomics Group. Really a wonderful team doing a lot of hard work in this space. Thank you very much.